The dipole polarity of the energy structures are being emanated by the subshell coding. The energy change their form of proteins or the electrons or protons. They change their shells by emanating themselves. They take the energies from the quantitative analysis. The qualitative value can be the three state of matter. The energies, the analytes which add to it make a energy shell membranes for the transfer of energy. So these waves can amidate the energy shells. So the next we move here, the analytes quantitative energies. Marobium value, here is the proliferative values for energies parabolical states. They can amidate in the metathesis reactions in our body, in the structural DNA progressions, and all the structures are being so the next is the gamino hydroxyl, which is the anionic value for transferring the cis complexity and the trans complexity phases. They make a teleomorphic phase for the energy subshell changes. So this is the complex molecule which transfer the energies from one domain to the another. They make an intercalary structure for transferring it. The tyrosine, the another is the tyrosinine, which help in the transfer of energy domains. So it is led by the oncogene. The lag reprocessor help in the bindation of from the gene expression and the energies of parabolical states. So the DAF2 is the energies paralyptical structure which make a intermediate structure with two mode of dipolar molecules. They make a catabolical state of matter for transferring the energy. Because of it, these energies are stable and can be used for anti-aging properties. Because of the reaction with the germline proteins, they make a strategic stochastic energies. So the dichloromercury is the states of dipoles which mediate their surroundings by coding and make a paralyptical structures of dipoles. They make an intercalary structure with each other and transfer the energy of protons, neutrons, and electrons for energy state of motion. So the hypothesis of hypos energy is the main hydrostate of matter. So the hypothesis is the hypothetical reaction which takes place in our brain structure, the psychometrical structure which is being made by it. The energy domains which changes hypothetically are known as the energy domains. So this is the energy domains which change from certain heights of second order to the third order of motion, third order of chemical kinetical equilibria. So next we will move here to the structure of qualitative analogy is made by medicines of homeopathic and biopathy of energy states of metabolical brain. So qualitative energies are being made by the medicines. They emanate themselves, as I have told now, that they emanate themselves by a structural process of analytical energies. So these energies are being mediated by the medicines. So the in homeopathic and biopathic of energy states, they changes the motion of energy, as the metabolical states did not take the states of atoms which are in outside the structures. They did not take that structure. So the structures will make a correct coding for transfer of energies, so that the metabolical states should take the energy. this happens with the homeopathic medicines. So, chemical composition required to isopathies of aging chemicals, which enriching the proteolytic of titrated energies of rotative of biochemicals. So the chemical composition is responsible for the amidating structure. We know that. So isopathies are the structures which make a energy with isometrical structure. They make a apolis, and all the structures are being dominated by it, by the structure of calculative quantitative analysis. We can calculate it by energy by making a biopathical energy is equal to amidations upon electrovalency. So the next we will move here could be the structures parabolical states. So the gravitic analysis would be here. So the gravitic analysis would be the amount which is being dissolved, and the two intercalary energies will meet with it and change their energy metabolical states to make a correct stoichiometrical uh, overlapping energies. The anionic bonding and the ionic bonding should be there. So the molarity, this changes the molarity, titrated changes and analytical changes are there. So the value which I have written here is the value of energies changes from high rates to low rates. The energy changes by the subshell membranes. These are the main things which are being used. The chemical structures are emanated by the dark DNA. Yes, the energies are being dominated by the dark DNA. So the dark DNA is a new part that can be discussed here. The dark DNA is a typical coded structure which comes from the variant variable structures of hypothetical reactions. It can be the value of quantitative analysis, which is the 3D. When we will calculate the value, it could be the energies paralyptical structure. So moving on the concentrated, this is the medicine which I have which the potential which I have told now. The potential changes in the energy states are due to the structure of it. These changes will make a potential of Gibbs free energy or the energy parabolical states. So moving on to the next is the fabricated etheric forms are concerned from high energy of hydration. The apolytical changes in the catabolical states of matter is dark matter like dark DNA. It has a coded structure which code the values of n states. The fabricated etherics are the energies which make a fabricated network of series like a Fibonacci numbers. They make a structure of their proteolytic energy. So the energy is coding can be the structure of it. So the hydration potential of the lattice energy is can be the apoplectical changes. So here is the cyclic hydrants which are being told now. So the mathematical reaction cycle states that states from polymorphic values. The polymorphic energies keep changing because of the many domains of p-n junction. They make apoplectical changes of lab processor or the protoplastide or the malate energies. They can make the different types of energies for transfer of oxidative positions. So, so this is the cyclic, quadric, energies, the cytokinetic fluid, which is the main part of it, which helps in the kinetical energies of prokinetical pairs, which make a hydro energies proliferations. The rest kinetical energies and the ubiquitin value can be the gravimetric energy. The gravimetric energy, as I told, the amount of energies which the biophysical energies have. So rest kinetical energy amidate it by making a proteolytical changes. So these are the hypothetical reactions which are given here. So if you like the video, please like it and comment it, and if you not subscribe it, please subscribe it, and please comment on this video to know what is the status.